Welcome to the deep dive. We've got this great stack of sources you sent over, and we're going to turn all that, dense, complex material into some clear, captivating insight.
And today we're really getting into one of the biggest questions in biology. Is it nature or is it nurture?
Right. Is your destiny sort of locked into your DNA, or is it shaped by the world around you? For a long time, the thinking was all about the DNA.
Oh, absolutely. The premise, especially after we sequenced the genome back in 2001, was, if we just read the whole book, we'd have all the answers. But that left this huge paradox.
The identical twin problem.
The identical twin problem. How can two people with 100% the exact same DNA go on to have totally different lives?
Yeah.
Sometimes, with completely different health outcomes. The rule book analogy it falls apart.
Because the rule book is only half the story. The really question you wanted us to explore is the problem in the genes themselves, the heredity, or is it the environment? And the answer in these sources is this amazing third option.
It's the layer above the genes. It's called epigenetics.
And that's where nature and nurture actually meet.
They merge. Yeah. I mean, to really get the core concept, let's use that classic metaphor. Think of your DNA, your genes, as the computer hardware.
Okay, fixed. Can't change it.
Exactly. The letters on the page. The epigenome, on the other hand, is the software. It's dynamic.
And the software tells the hardware what to do.
It does. It tells the hardware how to interpret the code. You're not changing the letters of a word, but you can, bold it or italicize it.
Yeah.
The word is the same, but how you read it, how it's expressed, changes completely.
That is the perfect mission for us today. We are going to dive into that software layer, how your environment, your diet, your choices can literally remodel that epigenetic landscape.
And not just for you, but for the legacy you pass on. And we have some genuinely brand new research to talk about, stuff that was published, just this week?
So to really get the software, we have to start with the hardware it's managing.
You do. The scale of the problem is just immense.
Right. So quick review. We've known since, 1944, Avery, McLeod, and McCarty, that genes are made of DNA. That's the blueprint. It's fixed.
But here's the problem nature had to solve. It's a packing problem. If you took the DNA from one of your cells and stretched it out, it would be about 2 meters long.
2 meters.
2 meters. And that has to fit inside a nucleus that's measured in micrometers. You can't just, ball it up and stuff it in.
That would just be genetic spaghetti. You'd never find anything.
Precisely. So nature came up with this brilliant solution. These little spool-like proteins called histones.
Yeah.
The DNA literally wraps around them, about 2 turns per spool.
Creating the structure. What's it called? Chromatin.
Chromatin.
Yeah.
It looks a bit like a string of pearls. But it's not just for storage. This is the key. The storage is the control mechanism.
So if you want to use a gene, you have to unspool it.
You have to loosen it up, yeah?
Yeah.
Open the chromatin. And if you want to silence a gene, you just clamp it down, make it super compact so nothing can get in to read it.
Okay, so how does the cell know which spool to loosen and which one to tighten?
And that is where the software comes in. These histone spools, they have these long tails that kind of stick out, and they're designed to have chemical signals attached to them.
Like little flags.
Little flags, exactly. A scientist named Vince Alfrey discovered this way back in 1964. He found that these tails get these chemical tags or modifications that basically tell the cell what to do.
So these tags are the molecular switches.
They are. Some tags signal for open chromatin-like, pulling a slinky apart. That means the gene is unholy. Other tags signal for compaction, and that shuts the gene open on F.
And there must be machinery to do this.
Yep. Specialized enzymes, we call them the writers when they're adding a tag, and the erasers when they're taking one off.
Okay, so you have the DNA hardware, the histone packaging, and the software of tags put on by writers and taken off by erasers. But what happens when that software gets a bug?
And that, right there, is the entire promise of epigenetics in medicine. See, if you get a true genetic error, like a mutation from smoking, that's hardware damage. It's permanent. It's really hard to fix.
But an epigenetic error, a mistake in one of the tags?
That's just a software bug. The underlying DNA is fine. And if it's a bug, well, that means it's potentially reversible. You don't need a new computer. You just need to reboot the system.
And we're actually seeing this play out in cancer treatment. A lot of those writers and erasers we're finding out are completely broken in human cancers.
They are. And that understanding is changing everything. The sources mention this patient at Memorial Sloan Kettering, larynx cancer that has spread to his lungs. He was treated with the first ever FDA-approved epigenetic drug.
And it worked by targeting those mechanisms.
Exactly. It was designed to fix the software bug, not attack the damaged hardware.
That specificity is amazing.
Yeah.
Let's talk about this discovery in pediatric cancers, the oncohistones. This just shows how depth this goes.
This is wild. In some really aggressive pediatric cancers, like certain bone cancers, the mutation rate is nearly 100%.
Mm.
But the mutation isn't in a normal gene, it's in the histone protein itself.
Wait. So the problem isn't the instructions, it's the actual spool that holds the instructions.
That's it. And the mutation is located right on the spot on the histone tail where one of those key epigenetic tags is supposed to attach. So this broken spool, the oncohistone, it physically blocks the writer enzymes from putting on the normal protective tags.
And that failure of packaging is what's actually driving the cancer.
It causes and drives it, yes,
Yeah.
which completely changes how you think about treating it. The goal isn't just to kill the cell, it's to maybe inhibit the inhibition, to fix the packaging so the cell behaves again.
So if there's hope for cancer, what about other devastating illnesses? The sources pointed to neurological disorders, specifically Rett syndrome.
Rett syndrome is a really severe autism spectrum disorder, mostly in girls, and it's caused by a mutation in a protein called MECP2. This protein is fascinating. It's an epigenetic reader.
A reader.
Yeah, it doesn't write or erase. It reads, it recognizes another key epigenetic mark methylated DNA and binds to it.
And this MECP2 protein is super important in the brain.
Incredibly, there are something like 17 million molecules of it in a single neuron nucleus. It's everywhere. And what it does is it acts as a bridge. It sees methylated DNA, and then it calls over a big gene silencing complex. So it's essential for keeping genes off immature brain cells.
The conventional wisdom, though, with brain disorders is that once the developmental damage is done, it's done. It's permanent.
And that's what makes this mouse experiment one of the most stunning findings in, like, the last 20 years.
Okay, what did they do?
They took mice that were engineered to lack MECP2. These mice were adults. They were already showing severe, measurable neurological symptoms. And then they turned the MECP2 gene back on.
In the adult sick mice?
Yes. And the symptoms they reversed.
No way.
Dramatically. The mice regained function. They lived much, much longer. And what this showed was that the problem wasn't necessarily irreversible damage done during development. It was an ongoing failure to sustain proper brain function because the silencing software was broken.
That completely flips the script. It
Yeah.
means that even years after symptoms start, if you can fix the software, you might be able to rescue the brain.
That's the profound hope here.
Yeah.
It's incredible.
It really is. So let's widen the lens from the cell to the outside world. James Watson, co-discoverer of DNA, he once said, you can inherit something beyond the DNA sequence.
And he was absolutely right.
Huh.
The human data is there. Just look at the Swedish famine study.
This was the one that looked at families in northern Sweden who went through these periods of feast and then
Yeah.
famine, right?
Exactly. And they tracked health outcomes, things like disease risk, weight, even mental health, across 3 generations. And the kicker was those outcomes didn't track with the family's genes. They tracked with the diet of the ancestor. Specifically, the father's diet during a critical period before puberty. The father's environment was being inherited.
And we're seeing this confirmed in animal studies, too.
Oh, yeah. There was a German study with fruit flies. They fed the fathers a high-sugar diet for just 2 days.
2 days.
That's it. And the next generation, their children, were obese. The researchers traced it back to epigenetic mistakes on key weight control genes that were passed down through the sperm.
That's an incredible level of sensitivity. And it's not just food, it's nurture, too.
Right. The work from the Mimi lab with mice, they looked at good moms, the ones who did a lot of extra licking and grooming. That nurturing behavior literally transmitted epigenetic marks that controlled the stress response in their children. The mom's behavior was setting the software for the baby's stress levels.
So, okay, diet, nurture. These things change the software that gets passed down, but the big question for all of us listening is, how long does that legacy stick around?
The data right now seems to point to about 2 to 3 generations. After that, these environmental marks tend to get diluted out. The sources say they lose their oomph through the big reprogramming cycles of reproduction.
Which brings us to the really personal part of this, the state of human sperm. The study comparing lean and obese men was a huge eye-opener.
It really was. They found that while the basic histone packaging was the same, the DNA methylation patterns, those little chemical flags, were profoundly different. And here's the crazy part. The genes that were marked differently were almost all linked to central nervous system development.
So genes for brain function and appetite control.
Master regulators of appetite, MC4R, FTO, the big ones. It's like the father's diet was pre-programming the child's future appetite through the sperm.
That gives men such a direct biological responsibility for their health choices.
It does. But here's the most important part of that whole study, the part that gives us agency. It's the dynamic response. They also studied morbidly obese men who underwent bariatric surgery.
Okay, a huge positive intervention.
A huge intervention. And they tracked their sperm epigenome before and after. And what they found was that it rapidly remodeled. Just 1 year after surgery, over 3,900 genes had changed their methylation status.
Wow. So it's not a permanent sentence.
Not at all.
Right.
It shows the system is vulnerable to stress, yes, but it's also highly capable of being reset by a positive change. You have the power to fix the software right now.
That is the aha moment. It's a repairable system.
Absolutely.
Okay, let's get to the newest research in your sources, the stuff published this week. This is where we go from just observing these effects to pinpointing the actual molecules that carry the memory across generations.
This is the absolute cutting edge, using germline epigenome editing in mice. So researchers could ask a very specific question. If we deliberately break the software in the sperm, what does the embryo do?
What software did they break?
They erase the DNA methylation marks at a very important spot, a region called H19DMR. An error there is linked to a human growth disorder.
So they created a known epigenetic mistake in the father's sperm. What happened to the kids, the F1 generation?
They had the growth problems, as expected. The error was transmitted, but, and this is key, when they looked at the next generation, the F2, the grandchildren, the problem was gone. It wasn't passed on again.
So the system corrected itself. But wait, you said the error was only partially transmitted to the kids? If they completely erased the mark in the sperm, why wasn't the effect in the F1s worse?
That's the million-dollar question. And that's the heart of the discovery. The mark was gone in the sperm, but somehow it was being partially recovered in the very early embryo, right after fertilization.
So something was remembering what was supposed to be there.
Some kind of epigenetic memory factor.
Yeah.
A secret anchor must have survived the huge biological cleanup that happens when sperm meets egg, and it was guiding the repair machinery.
It's like the embryo knew the software was corrupted and was trying to reinstall it from a backup.
A perfect analogy. And the team found the backup file. It's a specific histone mark called H3K9E3, trimethylation at lysine 9 of histone H3. They found that this mark was being placed on the paternal DNA, the one with the error, immediately after fertilization.
And why is that specific mark the perfect candidate for a memory anchor?
Because while most epigenetic marks get wiped clean during that big reprogramming event, H3K9E3 is known to be pretty resistant to that erasure. It's tough. It sticks around.
So it acts like a beacon.
It acts like a structural beacon, it survives the wipe, and then it guides the cell's machinery to come back and restore the proper DNA methylation. It's the bridge that ensures the correct settings are put back in place.
And how did they prove that was the bridge?
They used their editing tool to get rid of H3K9E3 in the zygote, and when they did that, the recovery failed. The growth defects in the F1 mice got much, much worse. That confirmed it. It's the critical memory factor.
That is just phenomenal. It gives us a precise target. So let's wrap this up. What are the big takeaways you want everyone to carry forward from this?
Okay, I think there are three. First, epigenetics is completely redefining what we think of as disease. That reversibility in the Rett Syndrome mice, it suggests that even complex neurological conditions might be fixable by just restoring the right software function. This is driving real drug development now.
Right. Second, the environment is a powerful architect of your legacy. Your diet, your choices, they rapidly change the epigenetic information you pass down through your sperm, especially for genes that control the brain and appetite. But crucially, it's dynamic. That bariatric surgery study shows you have agency.
And third, we're finally identifying the specific molecular bridges, like H3K9E3, that manage this epigenetic memory right after fertilization. We're moving from abstract ideas into precise, targetable molecules.
This knowledge really challenges that old idea that we're just prisoners of our DNA. We've seen a father's bad diet can impact his grandchildren, but we also saw that a powerful, positive change can reset that software very quickly. So here's the provocative thought for you to take away. If a negative exposure can set a future generation back, what deliberate positive choice could you make right now? With your diet, with your exercise, that could give a measurable positive epigenetic advantage to a future generation. What positive legacy will you choose to pass on?